Welcome to the tutorial. Here, in this video, we are going to explain the models of plasma membrane. But before that, don't forget to subscribe the channel and click on the bell icon to get all new notifications and keep up to date about all the new uploaded videos in this channel. Introduction. Before the advent of electron microscope, some biologists and researchers, for examples, Gorder and Grandel, Davson-Danielli, identified cell membranes using indirect evidences, and hypothesized different models, which gave us the basic idea of plasma membrane structure. After the invention of electron microscopy, some scientists, like J. David Robertson, Singer and Nicolson, contributed to the development of the modern membrane model. Here is the flowchart, showing the evolution and development of membrane models. Since late 19th century, a series of pioneering experiments were done by many scientists, and they also developed different membrane models over the years. Few such important ones are shown here. Here, we shall explain 4 primary models: Gorder and Grandel model, Davson-Danielli model, unit membrane model, and fluid mosaic model. Number 1. Gorder and Grandel model, 1925. Gorder and Grandel, the founding fathers of our current model of plasma membrane structure, proposed this model in 1925. Their prediction about plasma membrane was quite simple. According to them, the membrane was bilayer. So, its surface area should be half that occupied by all its amphipathic lipids, spread out in a monolayer. Two tests were carried out independently by them. Number 1. They measured the surface areas of different mammalian erythrocytes with the help of microscope. Number 2. They also extracted the lipids from erythrocyte membranes, from the samples used for measurements of cell surface areas, spread the lipids out at an air-saline interface, and measured the respective monolayer surface areas. They compared these two results and found the ratio of a cell and the monolayer surface area was of approximately 1:2 for a number of different mammalian RBCs. Thereby, they confirmed their bilayer model of the plasma membrane, with the polar hydrophilic heads facing outwards, that is, towards the aqueous environment on both sides of the membrane, and the nonpolar hydrophobic tails facing inwards, that is, away from the aqueous surroundings. Each layer of this bimolecular layer of lipid, around the cells, is about 25 angstrom thick. It is a schematic diagram of Gorder and Grandel model. There are some limitations of Gorder and Grandel model. Let's talk about that. Why wasn't the cell and monolayer ratio exactly 1:2? It is due to some of the incorrect experimental data, such as the miscalculation of the area and pressure of the lipid monolayer, and the incomplete lipid extraction. This model was also unable to describe the membrane function, and had false assumptions, such as that of plasma membranes consisting of only lipids. Even, this model could not define the things on surface tension, permeability, and the electric resistance of membranes. Though there are some limitations, this concept of Gorder and Grandel lipid bilayer model became the basic understanding about the plasma membrane structure. Number 2. Sandwich models. Or, protein-lipid bilayer protein hypothesis. There are two sandwich models, proposed by the scientists, Davson-Danielli and Robertson. First, we shall discuss about Davson-Danielli model. In 1935, Hugh Davson, a physiologist, and James Danielli, a chemist, modified the model, proposed by Gorder and Grandel, by including protein in the membrane structure. According to this model, a phospholipid bilayer lies between two layers of monomolecular globular proteins which are hydrated proteins. That means two layers of protein flanked a central lipid bilayer. It is trilaminar and lipoproteinous. There are two lipid protein layers, one facing the internal side of the cell and the other facing the external side of the cell. The lipid bilayer composed of phospholipids, hydrophobic tails inside, hydrophilic heads outside, and proteins coat the outer surface. As the lipid bilayer was sandwiched between two protein layers, it is called protein, lipid bilayer, protein, or sandwich model. Here proteins do not permeate the lipid bilayer. This is the schematic diagram of Davson-Danielli model. There are electrostatic interactions between the polar ends of each lipid molecule, and charged amino acid side chains of the polypeptides of the surface proteins. Other groups could be bound to the outer protein surface by either electrostatic or van der Waals bonds. According to this model, lipid bilayer would be about 6 nanometer in thickness, each of the protein layers of about 1 nanometer thickness, giving a total thickness of about 8 nanometer. In spite of being very thin, membrane is an effective barrier to the movement of certain substances. Such a membrane would be capable of distinguishing between molecules of different size and solubility properties, and also between ions of different charges. So, the cell membrane exhibits selective permeability. Evidence to support this model. When high-magnification electron micrograph of membrane became available, it appeared to show the evidence for this model. It shows a three-layered structure, shown in the picture, where a lighter-colored region can be observed. Between two dark parallel lines, proteins appear dark and phospholipids appear light, possibly indicating protein layers either side of a phospholipid core. Even the total thickness of the membranes turned out to be about 7.5 nanometer, which is very close to 8 nanometer, proposed by this model. Yes, there are also some limitations of the Davson-Danielli model. So, now we are going to analyze the falsification of the Davson-Danielli model, which ultimately led to the Singer-Nicolson model. While the arrangement of the phospholipids were correct, there was issue with the arrangement of the proteins. They are also amphipathic. Their nonpolar portions would block and separate the hydrophilic polar heads of the phospholipids from the aqueous external environment, causing the bilayer to dissolve. Experiments showed that these proteins were mobile, and not fixed in place, rather they could move and did not form a static layer, which was as per Davson-Danielli model. Experiments also revealed irregular rough surfaces within the membrane. So there were transmembrane proteins which were not solely localized to the outside of the membrane structure. The model also did not allow for the different structures and functions of membrane proteins. It means, the Davson-Danielli model is not only incorrect, but it is also impossible. B. Robertson model, or unit membrane model. In the late 1950s, electron microscopy provided additional information about the structure of the plasma membrane. In 1959, J. David Robertson proposed his famous unit membrane model after studying the cell membranes from electron micrographs of sectioned material. He concluded that the plasma membrane and the membranes of all cell organelles were similar in structure. According to him, the cell membrane is a trilaminar that is made of 3 layers: protein, lipid bilayer, protein. The physical appearance of this trilaminar model has led to the term unit membrane. We have already seen it in the earlier model, that there are 2 electron-dense zones, and 1 electron-transparent zone at the center in the electron micrograph of plasma membrane. The transparent lipid bilayer is surrounded on either side by dense proteins with a difference in type, for the outer and inner side. It means there are specific chemical differences between inner and outer side of the membrane. Whereas, there are same type of hydrated globular protein layers on both of the sides, according to Davson-Danielli model. In unit membrane model, the protein layers are asymmetrical and shows structural polarity. On the outer surface, there are conjugated protein and polysaccharides, that is mucoproteins. While on the inner surface, it is non-mucoid protein. Each of the 2 external hydrophilic protein layers is of about 2 nanometer, and the polar groups of phospholipids, of 1 layer of the bilayer, is of about 0.5 nanometer. So total external hydrophilic layer is about 2.5 nanometer thick. The middle layer is a hydrophobic bimolecular layer, made of phospholipids without the polar groups, is 2.5 to 3.5 nanometer in thickness. The total breadth of the cell membrane is 6.5 to 8.5 nanometer. Here were also some limitations of unit membrane model. These objections to this model were advanced during 1960s, which are as follows. Although there is no doubt about the similar electron microscopic appearance of nearly all membranes, this strict chemical interpretation of uniformity is no longer supportable. The unit membrane appears as a single trilaminar structure in plasma membrane, endoplasmic reticulum, Golgi complex, and lysosomes. But mitochondrial, plastid, and nuclear membranes are different from the plasma membrane, because their boundaries have an outer and an inner membrane, each with unit membrane structural plan. In light of these limitations, a new model was proposed by Seymour Singer and Garth Nicolson in 1972. Number 3. Singer and Nicolson model, or Fluid Mosaic Model. In 1972, Seymour Jonathan Singer and Garth L. Nicolson published the Fluid Mosaic Model of the structure of cell membranes. The Fluid Mosaic Model expanded on the Davson-Danielli model by including transmembrane proteins and eliminating the flanking protein layers that were not well supported by experimental evidence. This model remains the current accepted model of the plasma membrane. Here the arrangement of the phospholipids remains the same, but the location of proteins is different. The model shows that these assemblies are highly dynamic, that is, this arrangement is fluid, not solid. There are different types of molecules put together in cell membrane, and known as a mosaic. These molecules are constantly moving in 2 dimensions in a fluid fashion, similar to icebergs floating in the ocean. The movement of the mosaic of molecules makes it impossible to form a completely impenetrable barrier. According to the model, phospholipids can rotate, diffuse laterally, which happens very rapidly, or transversely, that is flip-flopping. This mosaic is of phospholipids, cholesterol, proteins, and carbohydrates, that gives the membrane a fluid character. Their proportions vary with the cell type. The plasma membrane is composed mainly of amphiphilic, having both of the parts, hydrophilic and hydrophobic, phospholipid molecules. Phospholipids are the main fabric of the membrane, primary major component. Cholesterols are attached between phospholipids as well as between the two phospholipid layers. Cholesterol is only found in animal cells. This model shows that proteins are embedded in the bilayer as integral, peripheral, and lipid-anchored proteins. The proteins that are integral to the membrane are a heterogeneous set of globular molecules, each arranged in an amphipathic structure, protruding the ionic and highly polar groups from the membrane into the aqueous phase, and the nonpolar groups, largely buried in the hydrophobic interior of the membrane. These globular molecules are partially embedded in the matrix of phospholipid bilayer, and may or may not penetrate through both layers. The bulk of the phospholipid is organized as a discontinuous, fluid bilayer, although a small fraction of the lipid may interact specifically with the membrane proteins. Integral proteins, integrins, are the second major component of plasma membranes. Peripheral proteins are located on the inner, or outer surface of the phospholipid bilayer, not embedded within the phospholipids. They have weaker and temporary connections, that is, they non-covalently link to the polar heads of phospholipids of the membrane. They are loosely attached on the surface of the membrane, and anchored with a few ionic bonds. Lipid-anchored proteins are located on the surface of the phospholipid bilayer, and are covalently attached to the lipid molecules embedded in the layer. There are some evidences to support the fluid mosaic model. Number 1. Phospholipid bilayer in membrane. The electron micrographs of plasma membrane show that there are two prominent dark lines which indicate the presence of phospholipid bilayer, whereas, it was wrongly identified in the sandwich model, that described the dark lines as protein layers, and lighter layer as lipid bilayer. The freeze fracture experiment also shows the evidence for phospholipid bilayer, because the fracture occurs here, along the lines of weakness, that is along the middle of phospholipid bilayer. Number 2. Mosaic-like structure. When biological membranes are freeze-fractured, there are some bumps, grooves and ridges seen inside the fracture plane of membranes. It is a mosaic-like structure consisting of a smooth matrix, but interrupted by a large number of particles. These particles are of uniform in size, 8.5 nanometer, for a particular membrane. The particles represent proteins within the membrane, has been suggested by recent experiments. The results of this freeze-etching experiments with membranes strongly suggest that a substantial amount of protein is deeply embedded in, not outside the phospholipid bilayer. Number 3. Membrane fluidity. Membrane fluidity means lateral diffusion, that is lateral movement of lipids and proteins found in the membrane. Experimental studies showed that integral membrane proteins diffuse at rates affected by the viscosity of the lipid bilayer in which they are embedded, and the molecules within the cell membrane are dynamic, rather than static. So, phospholipids and proteins can float around and even flip leaflets rarely. Lateral diffusion allows the distribution of new cell-synthesized lipids and proteins, membrane fusion, that is fusion of vesicles with organelles, separation of membranes during cell division, etc. Membrane fluidity can be demonstrated through two experiments. Number 1. Fluorescence recovery after photobleaching, FRAP. A small section of phospholipids is bleached and becomes distinguishable from the rest of the phospholipids. Over time, the bleached area will begin to disappear as the lipids begin to recover. This occurs because the lipids are laterally diffusing throughout the membrane, including the bleached area. Number 2. Differential surface antigen labeling and cell fusion. In this experiment, cells are taken from two different organisms, and thus, have different detectable surface antigens. Then they are forced to fuse. Now, one can detect that the antigens diffuse throughout the membrane. This indicates that the lipids are laterally diffusing through the membrane. All of these are possible because the membrane has fluid characteristic, so that it doesn't restrict their movement. This fluid mosaic model derived by the scientists Singer and Nicolson, still remains as the most accepted model that provides a fundamental understanding of the plasma membrane structure. This concludes the tutorial here. So, thank you very much guys for watching this video. If you like my video, please give a thumbs up below and share with friends, and don't forget to subscribe this channel. You may also want to watch my next video on the functions of the plasma membrane. You may find some other interesting videos in this channel as well.